Welcome everybody to the 39th edition of the Multi-Omics Reading Group. I'm your co-host for today, Cesar Miguel Valdez Cordova, and we're coming back from break with a very, very nice presentation titled Scalable Single-Cell Gene Expression Generation with Latent Diffusion Models by Giovanni Palla. And also, one of the authors, Jacob, is in the call as well. The call is actually pretty packed, so I think we're in for a pretty nice presentation today. Sinyu, I believe, will be reading a small introduction, so please, Sinyu, take it away as you often do.
Yeah, thanks, Cesar, for the nice introduction. Welcome, everyone, to our reading group today. So I'll introduce a speaker today. So Giovanni is a research scientist at BioHub at CZI, where he works on AI and machine learning applications for biology. He earns his PhD from Technical University of Munich and Munich. He is also a co-founder and co-developer of Assayverse, the open source ecosystem for single-cell data analysis. I believe everyone in this domain will know this package. So let's welcome him, and thanks for accepting our invitation. Yeah, it's all yours.
Thank you so much for having us and giving us the opportunity to present the work. So today I present work on a generative model for sequence genomics that we've been doing here at the BioHub, together with colleagues in Redwood City and Chicago. And indeed, as I mentioned before, Jacob, one of the authors, is also in the call. So I'll kick off by presenting a table of contents. So I want to give a brief overview on literature for transformer models and generative models in single-cell genomics that sort of motivated some of the architecture's decisions for our model. Then present model architecture and training details, and then I'm going through the experiments, ablations, and results. Feel free to stop me anytime if you have any questions and so on. And I'm not sure how you usually do it, but I'm happy to take questions during the call as well, during the talk. All right, so there is a lot of work done on transformer models for single-cell genomics, both in terms of representation learning and in terms of generative models or reconstruction models. They sort of divide, or a division like I wanted to present here is how they treat expression, because we typically, when we work with gene expression data, gene expression data consists of a table of cells in the rows by genes in columns, and in each entry in the table corresponds to an integer that is essentially it's a measure of how many times that gene has been measured inside a cell. That RNA molecule is measured inside a cell. A lot of work, though, is also looking at binarizing this expression. And in fact, by binarizing expression matrices, you recover a lot of the gene expression variation and cell state variation, simply because cell types often are defined by which genes are expressed rather than at which level. And so binary expression is an approach that has been used in the past. These are like sort of maybe models that are more, let's say, famous. Conformer, one of the precursors, you see. They use different learning objectives. Conformer is a mask language models. UC is a contrastive learning approach, and Search-to-Sentence is an autoregressive models where genes are encoded effectively just as tokens. There are also transformer models that look at continuous expressions, and by continuous I mean really like treating the data as count data. And this is an example, for example, in transcriptformer. In SGPT, some instances of SGPT, and then others instead use a different approach, which still uses a continuous expression, but have a type of contrastive learning, like the IDO-cell model or SC Foundation, which use a redepth-aware training objectives. And these models mostly look at representation learning, but also some of them show generation or reconstruction capabilities. A model that is not often mentioned, but I personally like quite a lot, and so I wanted to spotlight it here, is a model called SC-Mulan, which is an autoregressive models that in which the paper they actually really show generation capabilities. So they sample from the model, and it's an autoregressive model, so sampled by next token prediction. In this case, the tokens are pre-ordered and fixed order. And the thing is that indeed they show, when they show U maps of like true and generated data, it's a function of like in my opinion, the showcasing really that the model is able to sample well. Although you can see that there are some artifacts as well, and I want to point you to the paper for some experiments they run, but I wanted to highlight it here. There is also a lot of work on generating models for single-cell RNA-seq that are based on this approach of like latent diffusion. This is again a selection of those: SC diffusion, CFGen. These are models for synthetic data generations. Some of them are also CFGen, for example, is a multimodal model. CellFlow is also MLP based, or in fact also just using PCA as representations for perturbation prediction. And then SCVA-DR is also a MLP-based latent diffusion model. So all these models effectively use a very similar, or in fact pretty much the same approach that we employed in our paper. The difference though is that they're all based on like very, let's say, MLP-based type of architectures. So the autoencoder models are MLP-based, like SCDI. The set of genes is fixed at input and output, and they learn ex post flow matching models or diffusion models in the representations of the autoencoder. This is very similar to what we employ, but we thought that a limit of this type of work is that using the MLP as the architecture for the autoencoder is limiting in terms of performance effectively. As we're going to exploit like innovations in transformer models to really leverage transformers both at architectures for the autoencoder and the latent diffusion. So the motivation is that we really wanted to build like a powerful and flexible generative model. And the applications we had in mind are synthetic data generations. For example, to be able to increase or upsample specific cell types or cell states for prediction tasks. And also another one is perturbation prediction. This is maybe the most one of the tasks, the most interesting tasks in single-cell genomics currently. And of course, the generative models, perturbation prediction can be casted as a generative task, where you're interested in generating data in an unseen condition. We also wanted to be able to work with arbitrary number of genes and work with the representations of gene embedding. So also drawing from our work in transcriptformer, where we think it's a transcriptformer and other works like you see, for example, where they employ gene representations from protein language models that are particularly useful, especially in contexts where you're interested in doing cross-species integration. And in our case, for example, cross-species generation. We don't show any results on cross-species generation, but that's something we've been looking at. Furthermore, we wanted to have a fully transformer architectures, both at autoencoder and latent diffusion, for again scalability and performance. We wanted to have a fully generative model, and we also wanted to be able to use a flexible likelihood. So we didn't want to. We want our model to be able to sort of work both on normalized data, but also on the true count data. So really modeling the data as counts, which is commonly done in like SCVI type of models. So we went through, Jakob here is there is a question, yes.
Yeah, hi, I see, like you mentioned, you want to use the raw count as your like the data you want to deal with. So yes. I'm wondering, like, how do you think the model is going to be generalizable? Because like different data, like for single cell or for perturbation data, they the count they range dynamically, right? Like they are from different labs, different sequencing techniques, and how do you think, like, how do you deal with the problem?
Yeah, so first off, I think that using normalized counts will not solve the issues that you raised in terms of batch effects or artifacts or variations due to like technical variation due to like coming from different labs. Mm-hmm. The reason of using count data is that there are instances, I think, where you care about the full range effectively of gene expressions, which in normalized data essentially you remove by dividing by the library size. Actually, all the results we show here are based on true count data. Yeah, but again, it would be straightforward to change likelihood to a Gaussian likelihood and then use normalized data.
I see. Thanks.
And maybe if I could add something, that also one of the advantages of using a transformer architecture. So for example, the niche former paper, they had like technology and platform specific tokens, right, that they could introduce. So when there's the attention operation, it's like platform and technology aware. And they showed they were able to kind of leverage that to do some like batch correction and like modality and technology integration. So that could like another advantage here of your framework potentially.
Yeah. So yeah, we don't condition neither the autoencoders nor the flow matchings on this type of covariates, but it would be possible to do indeed, yes. I mean, we don't do it simply because the experiments we decided to run for showing the capability of these models don't require it. But in the CFGEM paper, for example, they show interesting results on integration where they can condition the flow matching models on these covariates and then remove the information effectively. So for a data integration perspective, and I think the results are very interesting. And I don't see how our approach would, I think our approach would also be able to do something very similar.
Yeah, exactly. And I had actually another question. I want to keep it for a bit later, but maybe if I could hint at that other question that also speaks to that point about the cross-species integration.
Yeah.
But I don't know if you were going to talk about that later. I know you said you weren't going to present results on that, but so for example, I don't know if you know about the Saturn work.
Yes.
To facilitate cross-species integration, they have like these protein, PLM embeddings, kind of form these metagenes, and that's supposed to really help in cross-species integration. So again, that's another way where, like your framework, from what I understand from the manuscript, could also kind of easily facilitate and enable. So anyways, that's maybe for further discussion. I don't want to break the flow.
No, definitely. That's something we're looking into now. We're interested in this approach because, like essentially, currently we use learnable embedding as gene embedding, but you could use embedding from protein language models, indeed like Saturn or like UC or like Transconformer, and use those representations as ways to translate effectively gene features across species, rather than using like homologous informations and homology terms. But because we use gene embedding, effectively it would be straightforward to add by just using those pre-trained representations.
Yeah, right on.
So I just want to say that this was our motivation. So again, I was very fortunate to work with Jakob, who is a generative model expert. In fact, I recommend you. He wrote a book called Deep Generative Models. I recommend you take a look. And so we literally went through generative approaches and decided to, based on the desiderata we had, to focus on autoencoder or latent diffusion models. So an autoencoder with a flow matching in the latent space. So this is the architecture. I'm going to walk you through the different building blocks, but just to provide you an idea, we have an autoencoder here, and that's trained in the first step. And then in the second step, we take the autoencoder pre-trained, and then we train a diffusion model, or in fact, we train a flow matching model using a diffusion transformer type of architecture. So a key architecture let's say ingredient that we used is cross-attention, and I just wanted to give you a very brief introduction, or no introduction, but like recap on cross-attention. So in self-attention, we have essentially from the input, we learn 3 different representations, like the key, query, and the values. And then with attention operations, we aggregate it and return an output, right? So cross-attention is effectively the same, except that for the queries, we use a different set of representations that don't come from the input, and enables to reduce or increase, essentially changes the context length of the transformer models in the attention operation. So in this case, the attention operation is the same, except for queries now use a different set of tokens effectively that don't come from the input. This is particularly useful because in vanilla attention, complexity is n squared, where n is the sequence length, of your input. So the number of tokens you have at input effectively. With cross-attention, with the query, for example, is much smaller. And here I use these numbers 30,000 and 128 because these are like some real numbers we use in the experiments where your input is 30,000 genes and we use a latent query of 128 latent tokens. You see that the complexity is decreased by a factor of 200 times. So this is very useful, first off, from a computational perspective, because you can reduce the attention operations by a big factor, both at input and output, effectively. We also think, honestly, provides a very good inductive bias. This is sort of like a bottleneck for reducing the representations that gene space, which we know is like highly redundant, and there is a lot of correlations in a lower dimension representation. There is lower dimension in the context length, though not in the embedded dimension. And so how do we implement this cross-attention? So the implementation is quite straightforward. In the encoder, we have an input, two input, in fact. We have gene embeddings, and we use learnable embeddings, which we index by the gene ID, effectively. And then we have counts. And we have an embedding layer that effectively mixes the two. And we try different things. In the paper, we have some ablations, but what we end up using is very straightforward concatenation. So we concatenate the counts with the gene embedding representations, and we pass it through a linear layer. And then the very first layer, since the gene embeddings and counts have been processed, it's a multi-head cross-attention block, which takes as queries a set of latent tokens, which is again much smaller than the original input genes, and squeezes effectively the sequence length to much smaller dimensions. Then we have transformer blocks, and then we have our encoder output. In the decoder, we have again transformer blocks, and then before the conditional likelihood, so before the last layer, we have another cross-attention blocks, where effectively the gene embeddings that we use as input are re-added as queries, as latent queries, with the cross-attention operations to essentially unpool the latent tokens into the original embedding or original gene space. So this approach of reusing the gene embedding for the output effectively is kind of like similar to the weight tying approach in like language models, like BERT type of models, I mean llama models also. In that case, the layer before the softmax, where you project the embeddings to the original vocabulary size, the output size, they are tied from input embedding to the output. Here, we are reusing the representation effectively. So we are reusing the embeddings at input for the unpooling operation cross-attention. Now you can see that you effectively can have like different numbers of genes at input and output, for example. We don't show experiments on this, but this is something that is possible to be doing because, we have an arbitrary number of genes that we can define at input or at output. So this is like, we have invariance. Please, Lila.
Sorry, I think I'm making you repeat a little bit, but what are you using to generate the gene embedding specifically? Because you show like a bunch of candidate models for doing so, but
the gene embedding is just like a dictionary. It's just a learnable embedding where we index with the gene ID effectively.
Okay. So you learn a set of embeddings directly as a set of parametrizable, like a matrix of vectors.
Each gene effectively is an embedding. Yes.
Yeah. Okay. So it's learned directly from the data. It's not you're not using another embedder model.
No, exactly. No,
it's
learned from the data, and that's, for example, what you would use for protein language models. Those embeddings would be replaced by the representations of a protein, for example. Indeed.
Okay. Thank you.
Yeah. So essentially, with this architecture, we then have invariance for the encoder, because of this latent aggregate cross-attention block with the latent tokens, we can have like arbitrary number of genes or shuffled, permuted, and learn the same, encode it in the same representations. But then in the decoder, we have equivariance with respect to the input. we use specifically, this is a, so we train on both cases. We observe that by just encoding the expressed genes, and so replacing the 0 genes with a mask token or a path token, stabilized training, and in fact improved slightly the performance. It's also cheaper because your context length then is not the total number of genes at input, but it's the total number of expressed genes per cell. But we think of this more as like a regularization approach, effectively, that improves the training, but it's not. I don't think it's just. I think it's probably like by tweaking the architecture differently, we would obtain the same result. At output, though, we use always all the genes. And that's because we want to compare with models that are not able to show these like arbitrary number of genes. So we want to show that we work in the same gene space. This is also, in fact, a choice to show that it's scalable, because I mean, doing attention on 30,000 genes or 30,000 features, it's quite expensive. But we show experiments where we get to that number of genes. But yeah, it could be used by doing an arbitrary number of genes effectively. And that's we're thinking about experiments now to show such capability. I think particularly in spatial transcriptomics, where you have, by definition of technology, fewer number of genes for spatial compared to single cell. So we thought that this was like a key innovation of our architecture, of like combining this idea of cross attention for the encoder-decoder model. And then I actually realized, I was talking with someone at NeurIPS, where a very, very similar architecture was used in a previous paper at a NeurIPS workshop in 2022 from Will Connell, where they have a very similar idea of using cross-attention for the encoder and then also for the decoder, reusing the gene embedding. In that case, though, they were interested in representational learning, and so they have a masked language model approach, and then use the embedding representations for things like data integrations and cell type classifications. I'll suggest you to look at the paper because it's quite interesting. What we use it instead is that we train this autoencoder model with cross-attention blocks for reconstruction, and then learn a flow matching in the latent space. And so this is done again in 2 stages. Yes, Sinu, please.
Yeah, sorry to interrupt you. Could you please go to the previous slide?
Yeah.
So here, so for the more detailed figure, I see that you only plot the like the masked ones, because you are doing mass modeling. Like you mask some expressed genes and predict those expressions. So I'm wondering, like, how is this way able to reconstruct all the expressions, or it's just used for compression? You just use the latent space, because essentially, Sorry. Yeah, essentially, if you do latent flow matching or diffusion model, you have to use the decoder of the VE to decode it back, right? So it has to be able to reconstruct all the gene expressions, including the expressed and also the non-expressed.
Yeah, so indeed, so actually, we use all the genes at output. So we always reconstruct all the genes. But at the input, at the encoder input, we only pass the expressed ones and mask the zeros, essentially. So it's not really masked language modeling in the sense that we are not only reconstructing the masked genes, but we actually reconstruct all the genes.
Yeah, maybe that's why I'm asking, because like if you, for example, if for gene A, it's expressed, and in the input, like for the encoder, it's not masked, and for the output, you're still predicting it. Then it's a very chill task for neural networks. It doesn't learn anything, right? Like, because you give it as input already. So that's why masked language modeling, they would, like, for example, they only predict those masked ones. They wouldn't predict those non-masked ones. So that's why I'm asking, because you mentioned like you want the VE to have the capability to reconstruct all the genes, but in the meantime, you only mask some part of the expressions when you're trying to put it into the encoder.
Can I jump in also, Giovanni?
Of course.
Yeah. So like, I think it's maybe good to think about it in this way, that we provide gene IDs and counts for these expressed genes. And then, like, implicitly, we tell our model that in the latent space, somehow take care of everything else, but we don't provide you any extra information. So we do this. Instead of masking, we should think about padding, that all these non-expressed genes are like discarded in some way in the encoder, though. But then in the decoder, we tell the model, provide me all the reconstructions for all the genes. So that's why it's like, at first we were, I personally, I was quite surprised that this model worked so well, because I was a bit hesitant that, okay, like not providing this information explicitly won't work, but actually it works. So it's somehow in the latent space, the model organizes everything in such a way that, you know, it contains the information about all the genes that are non-expressed. Because from the compression perspective, like, it's enough to say, okay, so these are only the expressed genes, everything else is 0. And so somehow the model learns it this way.
Oh yeah, that's interesting. Yeah, thank you.
And I think maybe it was, I don't know if it's directly inspired from the SC Foundation paper and the extremo gene architecture, where I think they do very similar thing, right? They only encode expressed genes and decode genome-wide, essentially, or like protein coding genes. So
yeah, isn't sorry.
Yeah, sorry, please go ahead.
No, I mean, SC Foundation and extremo genes, don't they use these like read depth aware loss, where they effectively like subsample the gene expressions and pass the input and output from this subsampling of the cell? So it's not strictly like expressed or not expressed. It's more like, it's kind of like getting it, learning a representation that is that removes these like sampling issues effectively.
But I think if you look at the kind of backbone architecture that they call extremo gene, I think in that work, it's more specifically in that manuscript, they simplified it to just like, well, kind of what you're suggesting here, right? Encoding express and decoding genome-wide. So I think in SC Foundation, they added like extra layers on top of that backbone. But yeah, I don't know, maybe, yeah, there's some overlap there, I think.
Yeah. Should I move on? I've seen you had a question.
Yeah, thank you.
Okay, so yeah, so let's go to the flow matching. It's so we basically use a very straightforward flow matching formulations with linear interpolations. So we learn a neural network here that regresses the vector field with an MSC loss, and yeah, just like this standard linear temporal flow matching. So we have we sample for noise, Z0, and data. Data is in this case a cell, and we build our interpolant and learn a vector field to regress that. We use an architecture called diffusion transformers. Since now we have latent tokens and not latent, not a vector valued representations, and the model takes also as input the time component, of course, and also additional a condition. So the condition here is dependent on the experiment. So for the synthetic degeneration experiments, the condition is things like cell type or tissue. For perturbation experiments, the condition is a combination of both the context, so cell line or cell types, and the perturbations, genetic perturbation of cytokines. So because of this, we use a classifier-free guidance, classifier-free guidance approach. The condition is also represented as a learnable embedding. And we have during sampling, we have a weight, and we show some experiments on varying this weight to sample effectively differently from the model. Yeah, I should say that this is like very like standard flow matching with linear interpolants. So we don't do anything, you know, very elaborate. But I think it's actually quite interesting. Would be quite interesting to extend this to use better or more complex sampling techniques. For essentially learning something from generative models better. So this kind of brings us to a reflection that we sort of stumbled upon by running the experiments, but it's something that is very it's a big line of research for latent diffusion models in image generation, which is essentially like looking at the representations, the latent representation of the autoencoder and the features of such representations. So in an autoencoder settings, we want to learn an approximate posterior given a prior, right? And there are all sorts of ways of encoding structure or prior informations in this approximate posterior. And in fact, actually, single-cell genomics is a field where this flexibility was very exploited a lot, right? There are a lot of models that look at disentanglement or conditioning representations for removing technical variations like batch effects. Think about like all the variants of SCDI. They all effectively are very similar in terms of encoder-decoder models, but then the structure of the later representation is very different and very dependent on the application of what they want to use. What's the purpose of using such later representations in the analysis? Here, our only purpose is to learn a later representations that can be used to train a later flow matching models on top. So it's kind of different, right? Because we're not interested in the structure of the representations for encoding prior knowledge or removing variations and so on and so forth, but really for making it as easy as possible for our flow matching models to be trained on top of it. And this is again, it's a huge line of research in imaging. These are papers that I had since the papers I was aware of in October or November when I prepared the presentation. But for example, just over Christmas, there were two other papers that look at different architecture choices and training choices of learning better latent diffusion models. One of the most common open source models for image generation called Flux from Black Forest Labs, they, again, like slightly before Christmas, released a very interesting blog post on their latest model. And effectively, the whole blog post is about really like this trade-off of like learning very powerful autoencoder models in terms of reconstruction, but then they also have to have a representation that is amenable for training up flow matching models or diffusion models on top of it. And this is sort of this concept of diffusability, so how easy it is to train the diffusion models. In the image representations, again, for the image applications, there is a lot of work on aligning the representations with self-supervised pre-trained models like Dino, for example. There is this line of work called representational alignment, and a lot of papers have come out of it. So yeah, I think it's interesting because for us, we also essentially, by running the experiments, we realized that there were like things that we had to take care of for where we would, have like a very good reconstruction model, like much better than the MLP models, but then we were not able to train a flow matching models, or the flow matching model was not as performant to train the ex-post. And this is also very data dependent, like how much data there is and the complexity of the data. So this is also dataset dependent. Although we have some we've been doing some experiments over Christmas where we try to like nail down the recipes and we find some common features. Please, Jakub.
Yeah, so also I want to add everything you said is totally true, of course. But I also want to highlight that there is a lot of work on AI for science in general using latent models, right? And this is also like really a lot of work. Oh, sorry.
Yeah, no, that's a great point. Yeah, I mean, models like La Proteina from NVIDIA or these all atoms transformers for molecules from FAIR, they all use like very similar approaches. And also they look at like this trade-off between reconstruction and generation capabilities. So yeah, just wanted to highlight this. All right, so, oh, yes.
Yeah, I'm not so sure whether you will discuss this later, but like recently, for example, the most advanced diffusion models from like Kamin or Science Group, like they are trying to do diffusion directly on the raw data space or on the like very powerful encoders latent space directly instead of relying on the VAE's latent space, because like the VAE's latent space are more Gaussian-like, and it might limit the capacity for the representations. So I'm wondering like whether you are also exploring this way or.
Yeah, that's a great point indeed. There is like a recent work from Simon Shi where indeed they show that they train, they use the sort of encoding representations from a dyno or self-supervised models, learn a decoder separately for reconstructing to pixel space, and then learning a flow matching, right, using that representation. Indeed, and then there are like very interesting experiments on how they narrow down the training architecture details. I think it will be very interesting. We're not looking at it, but I think it will be very interesting to look at take a self-supervised model, right, representation, and use the same type of approach of like learning a decoder separately and then a flow matching type separately for these. Yes, absolutely, yeah. Especially, yeah. The question is always like, what is the application, right? But I can see that in context again, maybe with like spatial transcriptomics, where you have arbitrary number of genes, you would benefit from. There is like recent work from my PhD lab on self-supervised representations for spatial and single-cell integration, for example. I think those are really would be very interesting application for such generative models.
Yeah, thanks.
Yeah, so what we ended up doing is that first we started with like a recipe that is common in the literature, which is using a very small KL, a small weight for the KL term in the variation autoencoder. We ended up actually most of the experiments running them without removing the KL and using a layer norm at encoder output and decoder input. So effectively, we still have like a sort of a constraint support by means of this layer norm at encoder output and decoder input, where the flow matching models it's trained on, but without relying on the KL term. Honestly, the results were like kind of similar. It's just like one less term to search for in training, but I think for our data wasn't like such a big. There wasn't like a lot of performance improvement. It was more just like, again, like simplify the training. One important thing though is that we do have to resize the embedding dimensions of the latent tokens at encoder output and decoder input. And that's actually quite important for the flow matching training. Again, we found out effectively that it's better to have like higher number of latent tokens, but smaller in embedding dimensions than like the opposite. So fewer latent tokens, but like higher in embedding dimensions. And this is not from a reconstruction capabilities, because actually for reconstruction, the latter is better, but from a generation capability. So for training the flow matching models ex post. All right, so I'm realizing that I'm only 15 minutes left and I haven't gotten to the experiments, but very quickly, likelihood we use a negative binomial. The parametrization of the negative binomial is effectively the same as SCDI. In fact, we can compare to SCDI directly. We can compare the likelihood directly with SCDI because we use exactly the same representations. The trick is that there is a library size term that multiplies the softmax output of the decoder. And this is important for generations because we had to sample from the library size. And for doing this, we simply sample from fitting a normal distribution on the 1D, on the histogram effectively of the library sizes per condition. But maybe there are other ways to do that as well that we haven't really explored. But for us, that approach worked quite well. And then we have an dispersion parameter that we learn per gene. Again, this is like literally out of a SCDI model. Yeah, so as I said, during sampling, we sample from the library size, and we don't sample from the histogram directly, but we sample from a normal, effectively like a Gaussian fitted on the histograms. I think it would be probably equivalent. Okay, so this is a little bit of training details. So we train the VAE for 400 to 600 epochs. We freeze the VAE, and then we train a flow matching with 100 to 200 epochs. Most experiments, we can run them on one H100, 80 gigabyte. We do multi-GPU training, but for a matter of like being able to run more experiments in shorter amount of time. The models actually are quite small in parameter sizes, at least for the generation, for the encoding, for the representation learning, the last, let's say, experiment we ran, they're bigger, but for the generation, they're actually quite small in terms of parameter sizes. Of course, they're larger in terms of flops, because we have this like large input and output cross-attention that makes it expensive. It's more expensive in terms of time and memory complexity. Actually, the latest results we are trying to update soon, they use even smaller models. Just as a comparison, the SCVI models and the SCFGen model we use for static generation, we compare to, are all like above 50 million parameters. So they're like literally like quite small. Do we use the following data sets? We have 3 data sets for the observational static data generation. We have 2 data sets for perturbations and 2 data sets for looking at for downstream classification tasks. So look at the representations learned by the model. They vary in terms of number of cells, number of genes, and number of cell types, or whatever condition you are interested in. I just want to highlight that we go to about 30,000 genes in the human lung cell atlas data set, and this is really like the key metrics in terms of scalability that we care of, being able to work on like large gene spaces effectively, and not having to predefine the number of variable genes. Especially in like foundation models for training like large collection of data where you don't want to do this a priori because it's all very data set dependent. For the perturbations though, we do decrease to 2,000 genes, highly variable genes, because that's what the baseline is also used. So we just do it for the sake of being comparable effectively. We use different metrics for generation. We use so for reconstruction we use likelihood, Pearson correlation coefficients, and mean square error. For generation we use the Wasserstein maximum discrepancy and Frechet distance. For perturbation specifically, we use the cell eval package, which implements various perturbation metrics. We use 4. 3 of them were the one used also in the virtual cell challenge. One of them is one that we select because we thought was particularly interesting. So it's overlap of differential expressed genes, Spearman, absolute error, and discrimination score. I'm going to skip the ablations, but I'm happy to share the slides so you can look at it later. Maybe very quickly, like this ablation here is on the latent aggregations. This is something we were asked by reviewers on using a different way of aggregating the latent token, aggregating the inputs. So instead of using like latent token cross attention, using standard like set transformer permutation invariant aggregation like max, min, and sum, and the latent tokens is superior in all cases effectively. We tried it on 2 data sets with different number of genes, and we look again at the reconstruction metrics, local likelihood, MSC, and PCC, and the embedding dimensions and models dimensions are all exactly the same. So it's all comparable. So in terms of reconstruction, on the observational data set, our model, the autoencoder model, is superior across metrics. Although, to be fair, for small data set like dentate gyros, which is less than 20,000 cells, it's comparable to SCVI. So, you see from a likelihood perspective, we're actually like similar to SCVI. But as the data set become larger and more complex, you see that the model, the improvement is quite boosted by a large factor. I should say that the NA here, we don't have standard error. It's because we didn't train like multiple autoencoders from SimGens. We took the checkpoints provided. So we don't effectively have like but it's a very similar SCVI model, so you should look at the SCVI models mostly. But yeah, just why we have the NA. In terms of generation, though, these are the 3 metrics. Here are 3 data sets. Again, on the smaller data sets, dentate gyros, we are, let's say, on par, better in some cases, but on par with CFGen. But as this become more complex, you can see there is quite a big jump in terms of performance across these 3 generative metrics compared to the other 2 baselines. We also look at marginal gene expression metrics. So this is the R squared per genes. So this is the R squares across averages across genes. This is the variance, and then the zeros is like effectively like the sparsity, whether we recover the sparsity. And you can see that actually we are like slightly worse than CFGen and Latent Diffusion in mean, but like this is probably like a very minor, basically comparable because it's very similar. In terms of variance, though, similar to CFGen, we're able to recapitulate very closely the variance of the genes, which is something that Latent Diffusion is not able to, and the same for the sparsity. So this is like a zoom in on one specific case we show in the paper. So this is the HLCA dataset, so the lung cell atlas. Now the data points are covered by generated red and gray true, and they are all these these are all clusters or cell types. So I don't have the color by cell type here, but imagine that these are clusters or color by cell type. So first off, the first thing is that you can appreciate from the UMAP, which is a bit qualitative result, but you can appreciate from the UMAP that our model provides a much better coverage overall of the various density. You can see that not always, right, but more often than the other baselines, we are able to cover the density and preserve though the small resolution clusters effectively, something that density diffusion, for example, is not. If we look at specific marker genes of a specific cell subpopulation here, these are alveolar cells, and then we look at the density of reconstructed genes compared to true. So the true expression is the gray, you see the dashed line, and the three colors of the three different models, so SCLDM, CFGen, and density diffusion. You can see that the model is able to, most of the times, better recover the marginal density of gene expressions. So here, for example, these are very lowly expressed genes, but with a long tail. You can see that CFGen and SCLDM they're similarly, maybe a little bit worse in this case, similarly recovering it. This is a COL1A and CFD are like marker genes that are more strongly expressed. This is again the density over all cells for that these clusters here. You can see that our model is recovering the main mode better than the others. We also look at the cross-attention scores, and because we think they could be like interesting for an interpretability perspective. Although this is like a bit initial results, so we're still looking into it. But effectively, what we do is that we compute an enrichment score on the cross-attention scores in both the encoder top and the decoder bottom, and then cluster these enrichment scores. And the enrichment scores are based on marker genes of the cell types. So the idea is that we would want to see that the cell types, the enrichment scores in the latent tokens, in the cross-attention for the latent tokens, both encoder and decoder cluster. And you can see that the pattern is different, but they broadly cluster the markers of cell type lineages. So let's say the neurons here, the orange and blue, are all clustered together. Again, this is not very convincing, but at least we can show that this could be like a means of interpreting these results. And I think here the next step would be to do it with changing the number of latent tokens to look at whether they would like essentially cluster the marker genes better, or other type of like enrichment scores on like pathways and so on. Yes.
Hi, yeah, just wondering. Yeah, this looks amazing. Like, I'm just wondering, like, whether it's possible. It's because like your model has those cell type or cell line as your condition labels. So that's why like the model can somehow learn more clearly about the gene relationship. Like, for example, the cell type marker with the cell types.
Yeah, that's
just
yes. Yeah, no, that's a great question. I should have said, so this is actually on the autoencoder, so it's not on the flow matching, and the autoencoder is not conditioned on anything. So yeah, there's no conditioning on cell types in this case. We do condition on cell type in the conditional generation setting. Yeah, so this is like purely from the model. And again, I think this is more of a function of like expressed genes, right? But I think interesting would be to do it on like other type of gene sets, for example, pathways activation and so on. But yeah. Okay, so second, like perturbation predictions, we use two datasets, the parts 1 million, which consist of PBMCs perturbed with cytokines, and then the reprogrammed dig is a standard genetic perturbation dataset. In this case, we have use only 2009 variable genes, and we condition. This is like the problem is like similar to what state does, right? So it's out of distribution combination of context and gene. So the model sees, and all the baseline as well, sees all genes and all cell lines or cell types. Sorry, all perturbations or cell line or cell types, but some combination of those are left out. In this case, for the parts 1 million, we leave out 27 cytokines from the T cells, CD4 T cells. And for reprogrammed dig, we leave out 372 genetic perturbations from the EPG2 cell line. So those perturbations are never seen in that cell line, but other perturbations are seen in that cell line, or the same perturbations are seen in the cell line. So we compare against SCVI, CPA, Cellflow, SGPT, and the state transition paper. So not the embedding, but the transition model. Comparing on generative metrics, so we do two. We use generative metrics to compare, but also we use the cell-eval metrics. So on generative metrics, interestingly, our model is on par with Cellflow, sometimes better, sometimes worse, both if you use all stochastically variable genes or if you use the differentially expressed genes. These are differentially expressed genes, so done per perturbation effectively. And again, you see that SCLDM is sometimes it's better for some metrics, sometimes it's worse. But I would say I don't think this is particularly. It's more for us. It's more to show that the model is comparably competitive to the in perturbation settings. Yes.
Hi, yes. Just wondering, like the setting for this perturbation task, because I know that for state paper, their holdouts for the PBMC, the parse data, and also the rep log, they are first hold out some cell types, and then within that cell types hold out some cytokines or genes. So I'm wondering, like, because you mentioned like you hold out 27 cytokines, but I think it's near like 60 or something for state here for PBMC data. So I'm just wondering whether your setting is different, like than the one presented in their paper, and you run their model again on your setting.
So these are all models that we all retrain these models. So we have the same split. That's a good point. Yeah. That's a good point about whether they are the same cytokines. I think in some of the experiments, I think they do use the same 27 cytokines. I will have to look it up though. But that's a good point that we should use the same set of cytokines. For genetic perturbation, we use a smaller set of perturbations. We use 300. Yes, but these models are retrained from scratch. So yeah, with the same seed, same split, and everything.
So just we use the setting, yeah.
Yeah.
No worries.
Yeah, no, that's a great question. So on generative metrics, I think it's hard to say, but again, our model is competitive, but not like overperforming. But using the other metrics, I think what's interesting is that our model is still like up there. So in some of these metrics, we are actually better than all of the baselines. But what's most interesting is that you see that like the performance of some of the baselines, for example, state for these metrics is much better. It's often on top or close to being on top compared to other models. Whereas before, Cellflow was like one of the top models, and now it's like not as close. So for us, essentially, show that we are competitive both in terms of the generation metrics, but also in terms of the cell-eval metrics. They also most of the times we outperform the perturb mean, so the average expressions for the perturbations, or the context mean. In this case, CD4 is the context because that's the cell that we leave out, or the FG2 context, that's the cell line that we leave out for a problem. And yeah, finally, we have this downstream classification task. So here, the idea is to train the model on a large corpus of, we use cell by gene human data, and then evaluate it on auto-distributions datasets. So this dataset, COVID-19, is a dataset of COVID infection, and then Tabula sapiens is a dataset of it's a cell type atlas. And these two datasets are left out from training for our models and all the other baselines we use. So they've never been seen during training. And here what we do is that we train the autoencoder model. Sorry, we train the autoencoder models, and then we perform linear probing evaluations on the representations of the encoder effectively. We train 3 different models with different parameter sizes. In the predicting cell types, the smaller model actually is close to being on top. It's on top, but again, here these are like very minor differences, so I wouldn't take these super seriously. But let's say again, the representation is comparable to other baselines we compare. In the COVID-19 infection, this is interesting because the largest model here is the top performing. Again, the differences are like very small. They are significant nonetheless, but quite small across different metrics. Yeah, again, to show that the autoencoder representation is nonetheless useful for as a representation, as effectively for downstream classification tasks. Yeah, so to conclude, we developed this architecture, which is a fully transformer-based latent diffusion models. We show performance in the generation task both in observation and perturbation data, and show that the embedding learned by the autoencoder is comparative with other foundation models. The future work we're interested in right now is understanding, again, like I said before, this feasibility problem, so the trade-off between reconstruction and generation. Working with different likelihoods, so working with normalized genes with Gaussian likelihood, and then also implementing more complex flow matching techniques for sampling, and then exploring multimodality applications. We have data sets internally on optical pool screen and single ceramic seekers, so we're interested developing models for multimodal perturbation prediction. And yeah, sorry I had to rush through, but thank you all for the attention. This is again the team, and yeah, happy to take any more questions if you have time.
Giovanni, thank you for the wonderful presentation. I do believe that will be spawning a lot of questions from the audience. I see a lot of hands that are being raised up now. So please, by all means, if you have a question for the amazing presenters, please do it now.
I see Philip. Yeah, Philip has his.
Philip, please take the floor.
Yeah, I'll start then. Thank you. Yeah, amazing presentation. I've been quiet the whole time, but mainly because it was very clear. But I do have one main question. So you said that you, in the end, opted for no KL regularization, right?
Yeah.
So technically, that means you move from a VAE to an autoencoder, and so you kind of lost that direct generative ability from just the autoencoder, sampling ability, let's say,
yeah.
from this autoencoder architecture.
But yeah. Sorry for interrupting, but not really even.
Okay.
Because like we lost the ability of sampling in the latent space, but not in the gene expression space. Because we still have the conditional likelihood function.
In the latent space? Or
no. Like in the gene, so the decoder is still, let's say, probabilistic.
Okay. Okay. Sure. But my question is, because I'm trying to figure out how much the flow matching contributes to the overall pipeline, and I wondered if you have tried maybe to have a small, still a small KL component, so that you stay in this VAE framework with a very good representation capability in the latent space. And what would have been the performances if you have sampled directly from the latent space using this VAE structure, rather than generating a latent point from this flow matching approach?
Yeah, this is a great question. This is a great question. So to the second point, we want to run the experiment now of sampling from the prior, in this case with like a KL regularization, and sampling directly from the prior. So that's something that we want to run now. But for the observational data, so the same settings for the comparison with CFGen and density diffusion, in the CFGen paper, they do have that experiments for SCVI. So they train SCVI and then sample from the prior, and show that the flow matching gives improved performance. I don't remember now the numbers, but I think it was significant. So I would remind you to that paper for, at least for the observational data. Yeah, but we should do it in these autoencoder settings, with this transformer-based autoencoder indeed.
And also to shed some more light on that, because like, first of all, thank you for this question, because it's actually very, very interesting. Like before we joined forces with Giovanni, I was playing around with small scale experiments, so much smaller architectures and also much smaller data sets. And I used like pure VAE, so no scaling of the KL divergence, the KL term. So it was a pure VAE. And then I played a lot with so-called priors. So Gaussian, mixture of Gaussians, right? And also like latent diffusion. And overall, there was, so what I noticed was the same trend as in any other modality. So Gaussian sucks, right? It's bad. Then with mixture of Gaussians, if you take more components, you're getting really good. Like you can really like model the latent space very nicely. This was still with non-tokenized latent space, so it was still like dense, let's say, right? So vectors. And then with latent diffusion, it was, of course, I was hitting the same like scores or maybe even better than mixture of Gaussians, right? So the trend was exactly the same. So given that, like when we joined forces with Giovanni, so we are very quickly, like we quickly moved to latent diffusion. We skipped like mixture of Gaussians, because also we had in mind this controlled generations, controlled sampling or guided sampling with perturbations, hopefully. So yeah, so like I hope this gives a little bit of context. Of course, it's a little bit harder to say how dense vectors, how these findings translate from dense vectors to tokens, right, in the latent space. But I can give my pinky that it would be exactly the same, right, or very similar.
Okay.
So Jakob, just to barge in for a bit, like I think that was spot on, and that sort of reminds me of like Stable Diffusion, right? Like you keep that sort of high resolution latent diffusion model, and that perceptual compression step, so to call it, like you're basically, would you agree or disagree with the next statement? Like you are relaxing regularization to ensure more sharp decoding in a way.
Yes.
Okay.
Exactly. I fully agree with that.
Okay, very good. No, that's a great takeaway. Thank you.
Thanks.
Oh, and by the way, any more questions, please?
Yeah. So there's a question in the chat. I guess I could read it out loud.
Yeah.
Hello. I'll read it quickly. Hello. I joined the. Okay. Small. I have a small question regarding expression prediction given multi-gene perturbations. Highly conceptually, how would you think this would scale using it as a world model to train RL for inferring, say, differentiation trajectories?
Yeah, good question. I don't have like super good intuition on how to train reinforcement learning models. I guess this will be done on like at the flow matching stage. Is that like to sort of like guided sampling, like with a pre-trained diffusion models, right? If you do guided sampling, I think it would be interesting for the differentiation trajectories, but I don't have a good intuition on how to do that.
So there's also sorry, Giovanni.
No, go ahead.
No, so it's so I think that if I understand it properly, so if we do care more about the latent space and then having a good reconstruction capabilities, so we also ran multiple experiments that we were able to actually get almost ideal reconstructions. But the price we had to pay was, of course, generative capabilities, because then it's like taking a bit larger latent space. It's harder to fit diffusion on top of that, right? So there's this trade-off. So I think that as a world model where we do care about like somehow compressing and then going back and then doing everything in the latent space. So I think this is, I would claim that actually it could be a good idea and then to use it within this framework.
Very nice. If there's nothing to add, we have another question by Boabang Francis. Please do speak up, Boabang.
I think he's muted.
Oh yeah, Boabang, you may be muted. Oh yeah, we got.
Oh, okay, I'm not from the biological background, I'm from the mathematical background, but I have a biological question. So the data you have out the COVID-19.
Boy, you're cutting out a little bit.
Yeah.
Can you write the question?
Hello?
Yeah.
Can you hear me?
Yes.
Yes.
Okay. So the data sets you had out, the permutation data set, was like all the data set, did they use the same kind of like screening mechanism, like let's say CRISPR interference, or they use like a different screening mechanism?
So the problem I think is a CRISPR is not interference is a knockout.
Okay.
They're not knockdowns, they're knockouts. These are like older generation, yeah. I think the modern ones are more knockdowns.
They are knockdowns, but were they the same for each data set?
The same technology, yes. So the problem consists of 4 datasets, 4 cell lines, and they were all the same, yes, all knockout.
Okay. So have you tried a scenario where you have like a different technology?
No, we've not tried that. No, I think honestly it would be very interesting to look at to do these in settings where you have knockdowns and knock up, so interference and activators. This is, I think, interesting from perturbation prediction in general, to be able to like model both down regulation and up regulations for the same set of genes. Yeah, but we have not tried that. I'm sure there are data sets. I think the Norman Adamson has that. Yeah, maybe some modern ones also.
So they were knockdowns, not okay. Thank you.
And also, like, maybe one thing, like, thank you very much, Bob Banks. It's very interesting. And also, like, to be completely honest, right? So, like what we did, in the guidance, right? So we used the categorical, also discrete variables, right? So it was like a finite set of perturbations. It's worth to highlight that we don't have this opportunity at the moment that we can go beyond perturbations that were unobserved, like, I don't know, chemicals, that were molecules, that, you know, like any molecule, right? So we don't have that. It's also maybe interesting to mention that it's like it's a bit unclear how this would behave, right, if it would generalize.
Yeah, maybe if the question is over, I would like to ask a new question. So is it okay to go back to the future work slides? Like, it'd be very interesting to hear, like, what do you plan to do for the next step for the these latent diffusion models?
Yeah. So one thing that we're still interested in is, again, understand better this trade-off between reconstruction and generations, like with discussion we had before. So this is effectively like running more experiments type of tasks, but I think it's important to like understand well this trade-off to be able to like train models that are more robust and so on. I think also likelihood and also sampling. So both essentially like different likelihoods for the autoencoder and different sampling techniques for the flow matching. And then multimodality, I think it's something that not on spatial atomics, but more on multimodal perturbation. But we are not working actively on this, but I think it would be quite interesting to work, to have this like sort of as a outlook.
Yeah. I would also reinforce that, like, I think it would be very interesting to look into this multi-omics, because, like, of course they are all connected. Some biologically they are obviously connected, but we cannot work in the same observable space because it's like comparing apples to oranges. So then having these latent spaces and somehow they interact with each other, and I don't know, looking at perturbation and at the gene expression level, and then trying to infer something at the I don't know, proteins level, right? So this is something that would require much more computational power, and also some sort of thinking how to combine all these ideas, because it's not, I think, straightforward from the probabilistic perspective, right? So this would be something that, for instance, I would be personally interested in. So then, okay, how to perturb one modality and then infer the other modality. That would be like nice to combine all these models. Because I think that for the modeling itself, what we have right now, I will sound super arrogant, but it's not my intention. I think that this is a very nice point that you can really start applying it and maybe like tweaking around it, but it's not like, I don't see like a huge jump, like, you know, in terms of architectures. It's more like secondary, right? It's like conceptually more interesting is, I think, to add some modalities.
Yes. Thank you.
How are you guys doing on time? Can you take more questions?
Yeah.
Or yeah, okay. So please, any more questions from the audience? Dylan.
Yeah, I was about to refrain myself, but you accepted to a bit extra time, so thanks for that. To kind of come back to, like, Philip had a question about, you know, the fact of removing KL kind of this simplifies the autoencoding, and you commented on the diffusability of latent spaces. That's not something that I've necessarily looked into a lot, so maybe you could, if you could spare a word on that, just generally, but also how you feel it's have you done like specific experiments to see if that had a large impact, or yeah, just your thoughts on the topic of diffusability of a latent space.
Yeah, maybe I can start, Jakub, and of
Yes,
course,
sure.
I'm sure you also have a lot to say. Maybe quickly, like, so the experience we ran in terms of like using or not scale divergence, they were effectively so modulo the same dimensions of latent representations, having the scale divergence or not having it, and sort of replacing it with a layer norm didn't give much differences effectively. In fact, I mean, the KL term is already very small, right? So it's kind of unclear how much it helps in terms of regularization. In one of the blog posts I linked in that slide, it's from Sander Dieleman. Let me get to it. Yeah, in this blog post here, there's latent diffusion models. It's they refer to it as like a vestigial aspect, because it's like it's there because we do variation autoencoder, but it's unclear how much is bringing it. On the diffusability side, I think a lot of work now in images is about learning a representation that is more amenable to training the diffusion models, and a lot of it is indeed, like Xinyu mentioned before, on like using representations learned with the self-supervised techniques, or even just using those, either aligning to those representations or using them really as like your encoder effectively. But I think, unfortunately, like a lot of that work, it gives indications for us or like for other domains sometimes, but a lot of it is also very focused on images. And so, you know, a lot of stuff is like about like spatial coherence and like preserving the locality and all these sorts of things, which in our data set, which is not, doesn't have a locality, right? Like we have an ordered set of genes. It's hard to say how much of those findings can translate to this application.
Does the I guess the theory or the concepts, are they related to why KL annealing was considered like kind of important? Like less now, I don't think, but before there's a bit like a debate about like maybe like let's say your decoder is a bit too strong, so then you're kind of hijacking the prior, so you want to anneal that process with KL beta annealing. Like, is that related at all to the idea of diffusability and the need or not, the lack of need for that KL term? Or maybe I'm mixing two topics. But
I think it is, but Jakub, maybe you wanna.
Yeah, no, so I think that in general, this is, yeah, I will give you a little bit of a shitty answer first, and then I will add something maybe that is interesting. It's like, I think in general, it is this very interesting interplay between the so-called reconstruction term and the KL term, right, in VAEs. And there's this old paper, Fixing a Broken Elbow.
yes,
Right, that it is very hard from the optimization perspective. It's super hard, right, because it's like one single objective, and then there will be always a tendency to improve either one term or the other term. And so I think this is also like here that there is this sweet spot for some specific size of the latent space for which the optimization works better overall. And so this is a little bit of a shitty answer in this terms that, well, it's again like this DL or AI magic, right? like you know it because you run a gazillion of experiments, and then you have
Right.
hunch. But maybe an interesting aspect is that at NeurIPS there was this one of the best papers, or like spotlights or something, orals or something like that. They had a paper on from INRIA from France. They had this paper on diffusion models, and they show that if you train diffusion models long enough, and they actually quantify that, so it's not like these mathematical terms like you don't know what it means. And then basically for like large diffusion, large denoisers, you can basically memorize all of your training data points, right? So
Right.
now it is something that how we train latent diffusion models, that we first fix the VAE or autoencoders, and then you fit a diffusion model to that. So basically what you learn, it's almost like learning Dirac's deltas, or like really Gaussians, right, around your latent points. So now it is like a little bit more obvious that if you fix your encoder, so the reconstruction term is now fixed, you don't change it. So now you can almost arbitrarily minimize the Kullback Leibler if you take large enough denoiser network.
So you're like memorization, but in the latent space with fixed representation. Yeah, I see.
Because we have this compression aspect, right? So then, of course, in the latent space, you almost memorize. So then the decoder has some errors. So that's why we see generalizability. So it could be that we are fooling ourselves in general, that these are like super good generative models, but in fact all the like generalizability comes from errors, right? But we don't understand that. So far as I know, the only thing that we really do understand is that yes, diffusion models can almost arbitrarily well fit any distribution.
Yeah, okay. So even like Dirac deltas on individual vectors or tokens in the latents. Yeah, interesting. Okay, excellent.
Sorry, so the last comment, because networks are smooth functions, so of course you can fit the Dirac's delta, so you always put a little bit of probability mass around.
Yeah, smooth it a little bit.
Exactly. So that's why you always have this like non-deterministic behavior.
Right. Well, let me say none of your answers were shitty, so thanks for those.
Yes, I wanted to barge in to say the exact same thing. I, for one, encourage the empirical crowd to run as many experiments if those are the kinds of answers we'll be getting post-talk. But yeah. All right. So I think in the interest of time, we should stop the session there, or the recording at the very least. But yeah, thank you guys for the wonderful presentation. Thank you for showing up, for staying for I don't know, all like 20 minutes over time. And yeah, that concludes the 39th edition of the Multiomics Reading Group. Thank you everybody for coming.